All right, let's go ahead and get started. Welcome everybody to the DCMB Seminar. Today's speaker is Dr. Yoon Lee from UNC. Dr. Yoon Lee is an associate professor in the Department of Genetics and Biostatistics at the University of North Carolina, Chapel Hill. She earned her PhD in biostatistics from here, the University of Michigan. And she had a very successful PhD career, including the top score on the biostatistics qualifying exam, which is quite an accomplishment, a Rackham Fellowship, and a dissertation fellowship. She started her position as assistant professor at UNC immediately after graduation, and she's been at Carolina ever since. And Yoon has published almost 200 peer-reviewed publications and was recently named a highly cited researcher by Thomson Reuters. And in her research, she develops and applies statistical approaches for genomic data, including analysis methods for genome-wide association studies, chromatin conformation data, and single-cell data, which I believe she's going to talk about today. So Yoon, thanks very much for speaking with us today, and we're excited for your talk.
Thanks so much for the very nice introduction, Josh, and really appreciate the invitation. You know, as a Michigan alums, I always feel extra honored to actually come back to share my research. And thanks so much again for the opportunity. So today, you know, as Josh mentioned, I will share with you our recent endeavors in the development of computational tools for single-cell RNA-seq data. And as you know, this is really an exciting and highly competitive field. I'm sure, you know, many of you have worked on, you know, such data one way or the other. You know, for example, I know Josh has several, you know, seminal pieces of work on this line of research. So the title of my talk today is Clustering Ensemble and Batch Effect Correction Methods for Single-Cell RNA-seq Data. So as the title suggests, I will talk about two important tasks in analyzing single-cell RNA-seq data, clustering and batch effect correction. For each, my team actually has developed two methods. And this is actually the first time, you know, attempt to talk about all four methods in one seminar. Likely a bad idea, we'll see. I'll try my best not to delay our dinner, and will deliberately skip some of the slides. And for some others, I will quickly scurry over, emphasizing only the key take-home messages. So my apologies in advance for those of you, you know, like who try to read the slides carefully. But three out of the four pieces of work that I'm going to talk about are already published. So feel free to read the corresponding papers and talk to me offline. I would be more than happy to discuss with you. So here we go. So as a background, you know, like
I'm sorry to interrupt. We're not seeing your slides.
Oh, I'm very sorry. Okay. Yeah, very sorry for that. Okay. So I skipped the. I just moved to the background slide. Now you can see it.
Yep.
Okay, good. Sorry. So as a very brief background, you know, single-cell RNA-seq data empowers us to dive deep into the cellular, you know, heterogeneity in complex tissues and during various biological processes. For those reasons, and because of the rapid, you know, technological advances, we have been witnessing really an explosion of single-cell RNA-seq data. You know, like the first study published, you know, barely a decade ago, only started with 8 cells. And then 8 years after, in 2017, you know, like from 10x Genomics alone, you know, it released over 1.3 million cells. And the volume and the complexity of single-cell RNA-seq data will only explode even further with many of the large-scale efforts to create a complete cell atlas for both humans and other organisms. So these are all very exciting, but as you know, like many of us know, the analysis of single-cell RNA-seq data poses many computational challenges. And as briefly mentioned, the previous slide will focus on two and talk to you about our solutions. So the two areas single cell directions I will focus on. First is like a clustering of single cells. So clustering is an essential step in defining the cell types from transcriptomic profiles. So the cell types are very important because they largely determine individual cells' identities and functions. So our group has published 2 papers in the past 2 years for Ensemble clustering of single cell RNA-seq data, so safe clustering and same clustering. And the second direction is batch effect correction. And it's very well known that batch effect is a plaguing issue in many omics data. Single cell RNA-seq data is no exception. As a matter of fact, as the single cell technologies evolve so rapidly, a lot of times, we have single cell RNA-seq data generated from multiple batches. For example, over a period of months or even years, and using very different capturing, single cell capturing or library proposition procedures with samples obtained under different experimental protocols. Or data generated by different labs, different technicians. So all of those can result in unwanted batch effect that can obscure biological signals. So for batch effect correction, we have proposed supervised mutual nearest neighbor based approaches. So we published the SMN earlier this year, and we have iterative SMN or ISMN method that under development that I will talk to you, show some preliminary data on. So as an outline, I will cover all 4 methods, 2 for cluster ensemble and the other 2 for batch effect correction. And as mentioned, I will focus on the key ideas and also focusing on the first method under each of the 2 series. So the first methods are in bold and underlined here. So let's start with the clustering example. So as mentioned earlier, clustering is such an important step in analysis of single cell RNA-seq data that many methods exist. For example, commonly used methods include Seurat, SE3, CIDR, SIMLR, SCAMP, SIMLR, just to name a few. And Josh actually also have a very widely used method called LIGAR and provide a rather elegant solution to this cell identification problem via transfer learning across multiple data sets. So there are many methods. And then you may ask, why do you even bother with yet another clustering method? So we actually have very good reasons. First of all, although there are many methods, when applied to various data sets, there are several methods that tend to perform reasonably well, but no method is a clear no-brainer type of winner. So more importantly, the existing methods differ sometimes rather drastically in their solutions. So the solutions are measured in terms of both of a cluster or cell type membership for the individual single cells, and in terms of the number of clusters estimated. So on the right-hand side of the slide, I show you results from 4 popular clustering method, the C3, CIDR, Seurat, t-SNE plus k-means on one data set. And the figure quantified the similarities of each pair of methods using 2 metrics, NMI and ARI. So NMI showing on the upper triangle, ARI showing on the lower triangle here. So both metrics measure similarity between 2 clustering solutions, and both range from 0 to 1, with 1 indicating complete agreement and 0 complete disagreement. As you can see from the plot here, these methods tend to agree, rather badly, with ARI or NMI really range from even below 0.2, and to the best is only around 0.6. And in addition, this method tend to vary in their estimates of the number of clusters. For example, in this data set, CIDR here says, okay, there are 3 clusters in this data set. SC3, by contrast, says, okay, there are 20 clusters. So Ensemble has been long recognized for its ability to make a more robust and accurate inference, and has been used a lot in the clustering, so-called more art than science clustering. So that's why we decided to explore building Ensemble solutions, which combine strengths of the existing methods. So that's the reason we're doing this. So we first build this safe clustering algorithm, and here here's the overall workflow of the method. We started the results from 4 individual clustering methods. So again, this SC3, CIDR, Seurat, t-SNE plus k-means. So this is the first step. So this table here provides a summary of the contributing methods. And we can see that they differ in how they deal with the dropout events and how they perform pre-clustering quality filtering, how they quantify between cell distance or similarity, and also in terms of their methods of choice for dimension reduction, for clustering per se, and for estimating the number of clusters in a data set. So I won't go into the details of the table, but want to point out that all those differences in all those aspects are largely responsible for different methods being the best choice for different data sets. And also for the same reasons, no method is a universal best choice across all the data sets. So again, that's why we build the Ensemble. So after we obtain, the solutions from these contributing methods, our safe clustering builds a hypergraph. So here's the hypergraph. For this toy example shown on this slide, we have 7 different single cells, and denoted by this axis here, x1, x2, x7, and then we have 4 different methods here, single contributing methods. So the first method, clusters the single cells into 3 groups of 3 clusters, with the first 3 cells going into the first cluster, and the next 2 cells in the second cluster, and the last 2 cells in the third cluster. And similarly for the other 3, methods. And with those, clustering results, solutions from the individual methods, again showing 4 individual methods here, and then we'll build this overall hypergraph on the right, with the number of rows being the number of single cells. So here again, there are 7 single cells, and the number of columns being the sum of the number of clusters from the contributing methods. So in this toy example, the first 3 methods each generates 3 clusters, and then the 4th method generates 2 clusters. So there's a total 11 hyper edges or 11 columns here. And then the individual value, the value in this hypergraph are binary variables, with one indicates, okay, this cell, for example here, this the single cell number 4 is classified into this second cluster of the first clustering method. This is how the hypergraph is constructed. And after the construction of the hypergraph, we attempted 3 different hypergraph partitioning methods, HGPA, MCLA, CSPA, which I will describe in a little bit detail. The first and simplest one, the hypergraph partitioning algorithm, or HGPA, is clusters the single cells, again, indicated by those. Seems to have a question. Oh, sorry. They are denoted by those axes here, again, X1, X2 through X7 for the 7 single cells. So HGPA clusters those single cells, which is very intuitive and straightforward. But this algorithm does not work very well in most of the data sets we benchmarked, as you will see later, because this algorithm is very sensitive to unbalanced sizes of individual clusters, which is very common because there are some common cell types with lots of single cells. They are relatively rare, like a cell types, for example, in brain, excited neurons, like oligodendrocyte, astrocyte, are all rather common, but like in contrast, like a microglia, like a parasite, those are relatively rare cell types. So because of that reason, like HGPA does not really work that well. So I'm not skipping the details of how exactly this is done, and we'll focus on this meta clustering algorithm or MCLA. We used the MATIS package for the implementation of MCLA. MCLA is a rather interesting algorithm, and it's like unlike HGPA, it is robust to unbalanced cluster sizes because it clusters MCLA clusters hyperedges of the columns shown here instead of clustering those single cells as HGPA does. Here again, the hyperedges corresponds. The hyperedges correspond to the clusters from the contributing methods, and while the vertices correspond to the single cells. So MCLA clusters hyperedges, while HGPA clusters the vertices. So that's the main difference, and that's actually one of the reasons, at least we believe, MCLA works well for single cell ensemble clustering. And then finally, we also attempted this CSPA, but I will skip the details because it didn't seem to work well either. And also it's computationally more intensive than the other two algorithms. So finally, after we get to the final clustering ensemble results, we evaluated the quality using the adjusted random index, which we already showed. So I'm showing you some of the detailed formula, but again, this is just a similarity index with one being, indicating fully green solutions, and 0 meaning the two cluster labels completely disagree. And we when comparing with the true cluster labels, true cell type labels, we prefer higher values. Then we benchmarked our methods in quite a number of data sets. We managed to assemble 12 different data sets, and they are of a different number of clusters, with them ranging from 3 clusters to more complex structure with 14 clusters, and the number of single cells ranging from below 50 to over 30,000. And those data sets also cover a wide variety of sequencing technologies, tissue of origin, data units. As you can see, some of them are quantified by UMI, some by counts, some by PKM, and technologies are very different. So we deliberately pick data sets that are more representative of various aspects of single cell RNA-seq data. And shown here, are the, again, ARIs for the different methods in each of the 12 different data sets. And from left to right, we have HC3, CIDR, Seurat, t-SNE plus k-means, the 4 individual contributing methods, and the rightmost is our safe method. So again, for ARI, the higher the better. So you can see across all the data sets, our safe clustering is either the best, in those cases, clear best, or close to the best. There's no data set where it seems to perform very badly. In contrast, and each of the individual methods seems to perform have one or some of data sets where it performs very poorly. For example, here, here. here, things like that, SC3 here. So it seems by ensembling, not very surprisingly, it provide a like a robust and accurate solution that achieves a very good balance. So this is in terms of the cluster membership for individual single cells, and we also evaluated the results in terms of the estimated number of clusters. So this is a scatter plot comparing the estimated number of clusters to the true number of clusters in the order again SC3 CIDR and also in corresponding colors. So the last panel maybe focus only on the last panel, which compares which shows the absolute distance from the number of cluster, from the true number of clustering defined by the corresponding papers. And here, of course, talking about distance to the truth, the smaller the better. And you can see, safe clustering seems to work the best. The other three methods seems to work reasonably well. I mean, although the variation seems to be a bit larger. SC3 is the worst because, I skipped the details, but if you stared at the table earlier, SC3, when inferring the number of clusters, used a distribution called the Tracy-Widom distribution that tended to overestimate the number of clusters when you have a large number of single cells. So as a quick conclusion, for the first method, we presented a safe clustering, which is an unsupervised ensemble method that provides a fast, accurate, and also flexible clustering for single-cell RNA-seq data. It has quite a robust and accurate results across a wide range of real data sets we benchmarked. And it was published last year, and the software is freely available via our GitHub. And then this year, we published the same clustering. So we used a mixture model to perform this ensemble task. So again, for this one, I will skip quite some slides, just focusing on the main idea. So in terms of the method workflow, it's similar to our safe clustering. We added a new method called SIMLR. So I'll skip the details here, but again, SIMLR actually differs from other methods as well in terms of how it calculates the distance between pairs of single cells. It actually uses also a different dimension reduction technique, and also for clustering and for estimating cluster membership. And then, so this is a part that distinguishes same clustering from safe clustering. So instead of hypergraph partitioning methods, SAME adopts a multinomial mixture model approach. So again, I will skip all the statistical, heavy slides. So I'm showing here just to show the method details, and also, just to show I'm not a charlatan. I'm not going to talk about all the formulas later in the afternoon. But here I'm just skipping those. But we introduce a hidden variable, which is the cluster membership in the Ensemble solution. Of course, we don't observe. It's what we want to infer, and we use the EM algorithm to specify the complete data likelihood after introducing the auxiliary of the hidden variable C. And the E step and M step are specified here. So again, like I said, I'm not going to bother with all the statistical details. It's published, so you can check out the details. I'm happy to share the slides. Compared to safe clustering, I added a few more data sets when doing the benchmarking, increased the total number of data sets to 15. Again, a wide range of technologies, organism of origin, tissue, number of single cells, and a similar number of clusters, and the quantification units. And just a quick summary of the results, and we added the SIMLR, and we also compared with our previous safe methods, and this shows the rank And the smaller the better. So safe basically compared to the other method is a very clear winner with the lowest or best rank across the various methods. So again, this is talking about the cluster membership for individual single cells. And then we similarly look at the estimated number of clusters. So you can see like, like I didn't show the absolute deviation, but like same again, even visually, you can clearly see like a same estimates the number of clusters most close to the truth, which is the x-axis here. And then interestingly, we also showed one, like an interesting finding based on our same clustering results. So when we applied same clustering to the 10x genomics PBMC peripheral blood mononuclear cells, we discovered a novel cluster. As some of you know, the 10x genomics PBMC data have a gold or silver standard cell type label for each of the single cell or for most of the single cells. And then, we basically, look at the cells that are labeled by the 10x as B cell, T cell, and also as NK cell. Very interestingly, it's for the NK cell by unsupervised clustering, ignoring the cell type labels provided by the 10x, same clustering identified a new cluster, which is the red here, separate from the other NK cells labeled by 10x. And then we look at the expression of two NK cell marker genes, the NKG7 and GNLY. And, as expected, the NK, most of the NK cells, they have a very high expression of those two genes. But the cells in the new cluster, same identified, seems to have a rather low. You can see, the median, for example, for the G7 marker gene is actually 0. In this new cluster of cells. And then we look at the cells further. So those are the single cells identified in this new cluster, and look at their profiles. So each row is a gene, and we group the gene according to marker genes of each of the 3 cell types. So the top genes are marker genes for B cells, and the middle genes are marker genes for regulatory T cells, and the bottom genes are marker genes for NK cells. And then on the left, we're showing, cells in quote-unquote normal clusters, meaning that unsupervised clustering from same generated, or clustered them in concordance with their true labels. So those cells are clustered separately from those cells and from those cells. So those B cells seem to express largely, the B cell marker genes, but not of the other, cell type marker genes. And similarly for T cells, and similarly for the regular quote normal NK cells. Again, those correspond to those blue green cells here. But interesting, if you look at the new, the single cells in the new cluster, they seem roughly falling into 3 groups. The first group, the left part, I mean, some of them express some of the NK marker genes, but definitely not as much as those quote normal NK cells. But you can see they also express the regulatory T cell genes, and in a sense, much more express those much more than what they should have expressed. And then for the second group, I mean, it's sort of they don't express any of the any group marker genes very well. And in the last group, express some of the NK cell markers, but they also express, some, regulatory T, but not as profound as in the first group. So this seems very interesting. I mean, of course, we haven't done any experiment to verify, morphological or functionally what they are doing. But this is, how I think, single cell clustering and those single cell data are useful to generate new hypothesis for experimental testing. So so here's I'll stop here for the same clustering. So again, like we used a more, like a statistical, more elegant way to perform clustering in Ensemble, this mixture model-based approach, and it seems to work quite well. And compared to same clustering, like the only drawback I would say is the computational cost is higher for same clustering than for safe clustering. So again, the work is published earlier this year with nucleic acid research, and the software is freely available. So now I'm going to, switch gears and talk about the second, part of on the batch effect correction. So I mean, if people have quick questions, I'm happy to take, but if not, I will just keep going. So as we know, the presence of a batch effect is really prevalent, and the batch effects are also known as technical biases across different samples, and they pose great challenges when we integrate data across batches, especially when the data are generated using different experimental protocols or from different labs. So batch effect actually will not only obscure the true underlying biology, but it can also generate lots of false positive findings. So in this toy example, there are two batches, batch 1, batch 2, each of them with 3 different cell types indicated by the C1, C2, C3 here. So again, here the different colors indicate the different batches. Red is from batch 1 and cyan is from batch 2, and the different clusters are in those different ovals. And you can see within each batch, the different cell types seems to cluster rather nicely, the ovals are largely separated from each other. However, when we naively, put all the cells together, although, those cell types still seem to, cluster together, but if you say draw a line here, the variation is driven more by cell type, by the color, than by, driven more by batch, indicated by power, than by cell types. So this simple cartoon illustration, shows the importance of performing batch effect correction. So because of for those reasons, batch effect, correction has been realized to be super important, and has become really a very essential pre-processing step when performing analysis integrating single-cell RNA-seq data across the batches. And there's a recently developed batch effect correction strategy that leverages information of mutual nearest neighbors, or MNNs. So this MNN, mutual nearest neighbor based strategy, has been adopted both by Seurat version 3 and another method proposed by Hypergraph et al. In a method called MNN, mutual nearest neighbor. And so this is a strategy that's realized, that has been recently recognized for its potential in terms of batch effect correction. So the idea of MNN, mutual nearest neighbor based approach, is to identify mutual nearest neighbors across batches. So in a cartoon illustration here, for example, in batch 1 we have 3 cell types here, and in batch 2 we have the same 3 cell types and yet a 4th cell type. And MNN basically tries to identify mutual nearest neighbors across the batches again, and use those information for batch effect correction, and then at the end generate a batch corrected gene expression profiles based on those mutual nearest neighbors. So this is a very sensible approach, but this approach makes a very important or rather strong assumption that the batch effect is actually orthogonal to biological differences. or at a very minimum, this strategy requires the variation due to batch effect are much smaller than those due to biological effects. However, this assumption is not always true in real data. I'll show you some example in quite a few of our own real data sets. Especially when those different batches were produced using very different experimental protocols. For example, I worked with the Duke investigator who used a smart-seq for one batch and then 10x for the second batch, more than a year apart. And also say when they are from different labs. And under the non-orthogonal scenario, such a global mutual nearest neighbor searching strategy actually may mismatch single cells from different clusters across the batches. So as shown in the cartoon illustration here, in batch 1, the cells in cluster 2 are mismatched to the cells belonging to cluster 1 in batch 2. So this is because, here's the batch effect dimension, and those are the real biological effects when they are misaligned. And when the batch effect is comparable to the biological effects, you may face this type of dilemma, and this mismatched mutual nearest neighbors would apparently lead to undesired correction results. So to solve this problem, to accommodate this potential issue, we present SMN, so supervised mutual nearest neighbor method that directly incorporates the cell type information for for batch effect correction. And with the help of the cell type information, SMNN restricts the MNN search within the same cell type instead of performing global search, ignoring the cell type labels. Of course, a lot of time we don't have the cell type information handy, but with clustering methods like the same safe and all the individual methods I mentioned in the first part of my talk, those cell types can be inferred via clustering method. There's many choices actually. And here's a framework of SMNN. We started with gene expression matrices here across the different batches, and we first perform single cell clustering within each batch. And then to obtain, accurate matching of the cluster labels across batches, so we have this cluster harmonization step here, and I'm not going into the details, but we have this step, and then afterwards, and we'll find mutual nearest neighbors within the corresponding clusters. That's where the supervised part kicks in, and with that, we'll compute the single cell specific correction vector, based on which the corrected expression matrices of the two batches will be derived. We first perform the simulation studies to evaluate the performance of our SMNN method in contrast to other methods, particularly the original the MNN method, which does not use cell type information. And in our so we largely adopted the framework of simulation presented in the MN method, but we actually all take a a different approach where we introduced the batch effect. We introduced the batch effect in the low-dimensional biological space. So the low-dimensional biological space in particular are the three-component Gaussian mixture, so as shown here. And then we project that into the high-dimensional space, mimicking the thousands or tens of thousands of genes in real single-cell RNA-seq data set. So the P is a projection matrix. And then, like I said, we introduced the batch effect in the lower dimensional space. And then, so here showing the simulation data is, like in the orthogonal case, where, like MN actually, like conceptually should perform very similarly to SMN. And here, we're showing the Frobenius norm. So the smaller the better, and you can see even in the orthogonal case, SMN seems to do also slightly better, but like it's reasonably close. But like for the non-orthogonal case, it's clear like like SMN outperforms MN by leveraging the cell type label information. And then we also benchmarked the method in several real data sets. It's summarized here, different, like technical platforms, different number of single cells, and different tissues, organism of origin. And then showing here is, one data, this is a UMAP plot for the, let's see, I think it's for the PBMC. Yeah, for the PBMC data set. And then this is incorrect results. So we have the 3 different cell types, the common myeloid progenitor cells, which can be differentiated into one of the 2 lineages, either granular macrophage progenitor cells or megakaryocyte erythroid progenitor cells. So those are the sublineages or offspring lineages from CMP. And you can see, like when we do and the solid field are batch 1, and the unfilled fields, those unfilled triangles are from batch 2. And as you see, like in our uncorrected results, it's there's clear batch effects, like with the batch 1 cells really at the middle, and then the batch 2 cells at the outside circle. And like when MN, after MN correction, you can clearly see, like the GMP cells and the CMP cells are clustered together. They are kind of mixed. You cannot no longer separate them. So it does a reasonable job, but like there's a lot of mixing of the cell types. Liger actually performs reasonable as well. It's just in a pretty weird shape. And the SMN visually seems to performs the best. And then we used the, like from the MANOVA, 2-way MANOVA F statistic to quantify the performance. So for that, it basically measures the within cell type across batches variation over the overall variation. So here, like the smaller the better, because we want the cells from the same cell type, regardless of which batch, to be tightly clustered and separate from the other cells. So SMN seems to perform the best on this data set. And just showing as an example. And then, like the data using, like the SMN corrected results, we also seem to identify differentially expressed genes that are truly, like more representative or retain biological features pertaining to their genomic function. For example, showing here, like SMN and MNN seems to identify DE genes with quite some overlap, but there are some SMN specific DEGs and some of the MN specific differentially expressed genes. And the GO-term analysis for the genes that are overexpressed in granulocyte, macrophage, progenitor cells are enriched with the immune regulation. So that makes, good sense because this is megakaryocyte, which eventually differentiate into platelet, and erythroid differentiate into red blood cells. So they are less involved in immune-related functions. While this granulocyte and neutrophils, eosinophils, like basophils, and also macrophages monocytes, those are apparently, like involved in immune regulation. And similarly for those genes, different, upregulated MEP versus GMP. I didn't show the results for the MNN specific genes, but like it's less obvious terms that come up from the analysis. So it's, like a relatively small set of genes, but like go terms that came up seem to make a lot of sense. So in the interest of time, I will skip this slide. I will also look at the reproducibility because a lot of times, like we don't know which ones are the truly differentially expressed genes. So we look at the, reproducibility identified from the two batches, and SMN seems to work better. I'll skip this slide. So as a quick summary, our SMN is a batch correction method for single-cell RNA-seq data. It explicitly considers the cell type label information to help identify mutually near neighbors. And then are more, powerful in terms of extract the desired neighbors within the same cell type. So based on the benchmarking real data sets, it seems MN, better mixes cells of all cell types across multiple batches, and also enhances the detection of differentially expressed genes between different cell types, and seems to more accurately retains or recovers cell structures. So it's published, earlier this year in briefings in bioinformatics, and again, software is freely available. So lastly, I will probably spend 2 or 3 minutes on our ongoing work. It's the iterative SMN refinement method for batch effect correction. The one of the issues with MN or with our SMN is like it searches for mutually near neighbors from the original expression matrices, which may not be accurate, particularly when the batch effect is large. The iterative SMN basically searches the MNN on the correct data, and then of course it's iterative because we search MNN and then correct the data, and then based on the corrected data we identify new sets of MNNs. And here showing again, it's a log F statistics, the lower the better. And we show, for different data sets when, the best performance is achieved. So for some of the data sets, particularly for this one, which is, more, it's the most complex among the 5 attempted, it needs like 3 rounds of MN search to reach the best performance. And you can see actually the performance is much better than just using 1 round of MN search. So, we applied the data to several, real data sets. And so I'm skipping the workflow. And we apply the data to our cardiac single-cell RNA-seq data, where we have 2 batches, again the same, like a legend, like a solid from one batch and open from another batch, and the different colors indicate the different cell types. So when applying ISM and you can see the cells seem to, after Seurat, it seems to cluster reasonably well. But if you look at the cluster batch membership, you see there's those places where Seurat seems to not merging the cells well. In contrast, SM and seems to merge data from the 2 batches reasonably well. So visually it's better mixing, and then in terms of the quantitatively, we again use the F statistic is substantially reduced F measure. So again, like I said, we quantified in several real data sets, and as iterative SMN seems to perform, very well. And then taking the data, we also, identify differentially expressed genes, and the gene. So compared with the Seurat, the genes that the DE genes identified by both methods, of course, they show very strong. Again, this is the cardiac single cell. So we look at the cardiac mouse cardiomyocyte versus endothelial cells. And the two different types of cardiomyocyte, it's not really two different types, it's just a difference in their mitochondria percentage. Anyways, so the common genes show very strong, cell type specificity, like with the cardiac specific genes express, genes that are marker genes are very consistently highly expressed in cardiomyocyte. And in contrast, the genes that are specifically identified by iterative ISMN seems to work pretty well. I mean, like it's a little bit noisier than the shared genes, but reasonably well. In contrast, if you look at the Seurat specific genes, only about 70% of those are found to be upregulated in CM, in contrast to ours, close to 95%. And I would, point your attention to this particular CRY-AB gene, which we, followed up by IHC validation, and we performed, IHC staining for this CRY-AB gene that we identified based on ISMN corrected results, and also two marker genes, one for cardiomyocyte, the other for endothelial cells. So this is the cardiomyocyte marker gene. This is an endothelial marker. And this one is the gene that we are trying to test, the CRY-AB, and you can clearly see this is indeed a cardiomyocyte specific gene. We basically validated our results that specifically identified by after ISMN correction. So in the interest of time, I will skip this one as well. And so as a summary, we updated this our previous MNN supervised MNN approach into an iterative manner that seems to even further improve the performance. So as a final words, this is really a very exciting area of research, and there's still lots of room for improvement. For example, how to scale up with millions of single cells. I think Josh has some very exciting work along this direction. How to deal with multiple subjects. We have published, we just recently published another paper that performs multi-sample, differential expression, treating, the single cells from multiple individuals as repeated measures, and also how to do iterative, analysis when incorporating other omics data. And Josh Liger actually does a good job along this line of research. So with that, I'd like to thank many people, contributed to the work, specifically Chen Yang, former postdoc, now faculty member at UNC, and a former student Ruth, and current student Gang, and lots of collaborators at UNC and other places, and also folks in my lab. And thank you very much for your attention.
Great, thanks June for a great talk. And please feel free to ask questions now, either in the chat, or you can unmute yourself and ask your question directly. And I see we already have a question in the chat here.
Okay, see.
All right, I'll go ahead and read the question in the chat from Dana King. Thank you for a compelling talk. Perhaps this is out of scope, but I'm curious if simulations or test data sets for SMNN or ISMNN also investigate the impact of pre-filtering the data. How much should we be considering aspects like percent mitochondrial gene for our individual data sets, especially if phenotypes like cell death can have technical sources from generation of single cell samples or be biologically relevant, such as during developmental defects?
Yeah, this is an excellent question. So the short answer is no, we have not, use the simulation. In a sense, very hard to design simulations to answer, questions on the impact of pre-filtering of the data or different levels of QC on the data. But particularly for our SAME and SAFE clustering in our software implementation, we explicitly allow users to tweak various filtering criteria criteria, like the number of transcripts or genes expressed in the cell to perform cell level filtering, and also a percent of mitochondrial gene. And in a sense, like one of the reasons, like I motivated the development of this Ensemble method using by talking about the discrepancy of existing method. But the other thing, actually, when I started designing this, is like this Ensemble method can totally be also applied to like a different filtering of the raw data, and you can build the Ensemble solutions from those like a various level of QC on the data as well. Not to say like we should take cells with only 20 genes expressed, but there's a reasonable range, whether you allow 5% mitochondrial genes or whether you allow 10%. Both are very reasonable. And actually for like our cardiac single cell, you may notice, like as I explained quickly, our CM1 and CM2 are different in terms of the percentage of mitochondrial genes. And we initially used for the CM2 where we actually used a very stringent, I can't remember, 5% or 1% mitochondrial gene maximum. But then, like since the cardiomyocytes are actually very, like energy dependent, so it's possible that like they do express a larger percentage of mitochondrial genes. So we ended up including some more cells by like loosening the criteria. Yeah, this is a good question, but like no, we haven't done any systematic evaluation of those pre-filtering of the data.
Any other questions for Yoon? I actually have a question. So one of the real challenges here is determining what ground truth is when you're clustering cells, and also experimentally validating those clusters. Do you have any thoughts about what are the best ways to find these gold standard labels or validate your results experimentally?
Yeah, that's an excellent question. Actually, like that's one of the places we struggled a lot with the, like with both manuscripts. I mean, like even if we know the truth, let's, like in a lot of cases we're talking about gold standard or silver standard, like based on marker genes or based on, factor sorting. If we started with, like cells, where we sort of know which cell type they come from. But a lot of time, even with, say if we have the true, like expressions, not considering all the noise data from single cell RNA-seq, like if we apply, clustering, for example, hierarchical, there's really, a line we need to draw. Are we talking about just helper T cells? Are we talking about, like TH, 17 and TH, there's different types of helper T cells, and there's different, going up, there's different, T cells, there are different immune cells. So, like whether, for the PBMC data set, I didn't, like talk too much, but like we have, like a challenging case where we're looking at 3 different types of T cells, and we have a simple case where we're just looking at, B cell, T cell, and NK cells. So in a sense, like where you draw the lines, are you looking at the major, cell types, or are you looking at subtypes? I think that makes a big difference. So I think, like to answer your question more directly, so I'm talking about those. There are several ways. First is, like we, should be a bit open-minded to accept, like a silver standard or even, like a, bronze standard truth, so that, we can have a rough idea regarding, how well those methods perform. But like we should also, like the example I mentioned in same clustering, if we identify interesting clusters or seemingly new biology, I think experimental follow-up would be useful. And then similarly for the ISMN, we find this CRY-AB gene that we didn't know as a, like a cardiomyocyte specific gene. So yeah, so I think this single cell RNA-seq, has been really used more for hypothesis generating rather than, confirming. So I think, we need to be careful, use as much existing data to make sure that we don't dive into, expensive experiments without, first checking to see the results might be consistent or, conflicting what we know. But if there's not enough data, which is a lot of time the case, but we are observing, at least a consistent, evidences for multiple single cell RNA-seq data sets. I think it's where, we prioritize our hypothesis generated from those single cell data and go from there. So I don't know, I've been being hand waving, so I'm not sure I directly answered your question.
Yeah, those are good thoughts. It's definitely a challenging aspect of single cell analysis. Yeah. Great. Well, we are out of time. So thanks again, Yoon, for a great talk.
Thank you. Thank you so much.
And yeah. And so everybody, hope to see many of you back next week for next week's seminar. Take care.
Thanks a lot again. Bye.